Hello, good morning, good evening. Thanks for listening. This is a medical cannabis show. I respect the law and I hope you do too. And this is programs for education and scientific purposes. So I like to fact check my information and show university slides that are relevant to the topic. And I condone all recreational drugs and that type of thing. And today is an open topic, so nothing's been organised. So hopefully someone in the chat will give a cool topic or a discussion point again, and we can go from there. And if you're watching this back and it's gone for 10 minutes, that means there was no real discussion today and it was a short show. Or if you're watching back and you want to skip to the best, like to when we start getting into the facts or what the questions are, just really skip a bit and just look for the slides, I suppose. That's the best way I can suggest. And it usually happens between, I don't know, 3 to 10 minutes. That's when the topic will kind of start. And a few people here. How you going, 420 Fred? G'day to Shipwreck. How's it going? And Sunshine. RB. Hello, Sunshine. So, yeah, I'll give another 15 seconds and think about something to say. Not much has come to mind. The 15 seconds is up. It's 2 minutes now. And I still not real sure on what to talk about. It hasn't been a short show for a while. I say that for a bit now because it hasn't been. And there's been a couple of 2-hour streams, which is usually longer than the 1-hour stream that I like to do. That's okay. Must have been a good topic, hey? So have anybody heard anything interesting of late, or any myths or anything like that they thought they might want elaboration on? Or what else could there be? I don't really want to talk about me because this show's more safe for everybody else. So wait for something to happen. If not much happens, looks like it's not much happening. I've still got to give it at least 7 more minutes. I'm going to try. What can I talk about for 7 minutes then? The solstice is coming soon. The weather's always a good topic. You know, that's one of the most talked about topics in the world. So let's discuss that possibly. How is the planet going at this point in time? Well, we've just passed really the peak middle of the winter for the southern hemisphere and the northern hemisphere summer. So it's transitioning, but it takes 3 months. No, it's temperature has a lagging effect. So by the time it happens, you get the solstice, then it's 3 months usually from there. That's how the planet's done it. In Australia, they've done it a bit differently. They've set it at the start of, they've sort of shortened it by a few weeks, bring it back to the start of the month and whenever the solstice or equinoxes are happening. But yes, what Shipwreck say, it has been a very wet summer here. Oh, that's wet summers aren't really good. Doesn't that breeding all of the mosquitoes and stuff like that? Is that how that works? I know in the Northern Territory it used to be something along those lines. What was it when there was a really wet summer? The mosquitoes could lay all their larvae, so the following season, look out. And in the Yukon too, Holy moly, mosquito attack. We even had a couple of tornado warnings. Oh, full on. Jeez. Hope you don't get any anything lifting up your garden. The mosquitoes have been really bad here. Yeah, that's no good. Do you know that here's something interesting with plant science and mosquitoes? If I'm playing around with a spirulina bacteria, Arthrospira platensis, and by putting that in water outside, mosquito larvae don't multiply. So for my pets, I tip a little bit of that in, and there's no ever any larvae or anything to produce in it. I don't know how it would go large scale because I think it would take over. So I wouldn't be putting it into a dam or a river or anything like that. But if you can contain the water source, it's pretty cool. It's just something about mosquito larvae and plant science. Sunshine says humidity is high in Melbourne. Oh, yuck. Humidity. You can't, it's so, aeration's about the only thing you can combat with humidity. It's putrid. Dehumidifiers. I love the dehumidifier. It adds a little bit of heat also in winter when it's usually a bit more moister because the temperature drops and relative to the humidity. So when the temperature drops, there's more moisture in the air. So in winter when it's a bit colder, you generally get that. So having your dehumidifier, it can add a little bit of heat too. It's pretty cool. Humidity is high in Melbourne. Yeah, that's no good. Shipwreck says that's good to know mosquitoes are so bad. So bad. Yeah, I had the same culture for a couple of years now, and it's called spirulina. If you look on Wiki, actually, I'll show you. It's just a spin out. You won't believe how good how nutritional it is. I'll just share the screen. So for those who are into health and plants and soil science, this is a good one for both. It's good to know mosquitoes are bad. All right, I'll get rid of that, and I'll show you what spirulina. I can't believe we're talking about this. But yes, they used to call it blue-green algae, but it's not algae because it's a eukaryotic cell. No, prokaryotic cell, sorry. Algae is a eukaryotic cell. So it's a bacteria, but it is a bacteria. But there's three species. That's the one I've got. And the other two are nitrogen fixing. So unfortunately, I got the wrong type, but as for, so I'd be sourcing these two if I was going to be getting it again. I've had this one for a few years. But let's go down and just quickly, spirulina thrives. You don't need to do any of that. It loves CO2. I leave it outside. My batch culture. Food and nutrition. No, come on. I'm just going to briefly show you what it's good for. Oh, really? They haven't summarized it. I was hoping to say it's good for this and this and this because it does so much benefits for the human body. Food and nutrition. Spirulina has been investigated to address food security and malnutrition as a dietary support. It's top dollar when you want to go to the health store and buy it. So you dry it out, you scoop it off the top. It'll create a film, and you scoop it off the top and then dry it. And then it's got amazing nutritional. And for plants too, it contains a high amount of tryptophan, I think. No, that's the wrong word. But it's a precursor to auxin. So it's really good for plants. But it does everything. It's a superfood. This is really, really poorly explained, sorry. Because I thought it was going to... Spirulina has many adverse interactions. Oh, yeah. Toxicology, yeah, really good. Well, there was somewhere where I read where it said it was just rad for this and rad for that. It boosted the brain. It did all these types of things. It's probably up the top where I just skimmed over. But it's very, yeah. Hey, it's raptor. Mosquitoes. Oh, yeah. Sancho says, I am interested in your preferred bloom amendments, preparations. Bloom, top dress. You should have already made your batch up. When you've done, put your when you transplanted, and if you didn't transplant, you should have made your substrate up really, really well, healthily, with all your different amendments that you want in it. You'd know if, like, Pink DB is a potassium pig and usually gets potassium deficiencies, so you'd up it a bit in that area. I'm interested in your preferred bloom amendments preparations. Bloom amendments. Well, top dress. I like the top dress and have the right microbes around to unlock the elements that are immobilized. I don't know if you want me to go right into it. There's a few more things in chat. Let me know if you want me to go a bit more into it, because it's a very sort of a broadish question, Sunshine. I'm not sure if I answered it though. Shipwreck says, I remember back in my big outdoor days, I would look over at my buddy who was helping out, and there would be a haze of mosquitoes around him. Yeah, yuck. Yeah, that's after the wet. Heaps of that's what happened in the top end. Heaps of, like, monsoonal rains allows that to happen, because they need somewhere to breed. You know how they breed, like any type of blooming water. G'day, Raptor. Nice to see you, mate. Shipwreck says, I was almost having a penny tack. How many years around us? Yeah, yuck. You need those head nets. They're pretty cool. I've never worn one, but if I was under attack again, I've needed them in a few situations. But if I was under attack again, I'd be getting them. So you've seen them. They're like bee nets, but they're for mosquitoes and stuff, and you can see through it and stuff. I'd get a better one, so it lasts at least a few seasons. Hey, Genetic Memory Farm. It's nice to see you, mate. Well, need to make some lactobacillus serum based with spirulina. Oh, see, that might be an example of two bacteria that aren't symbiotic, because, you know, of the 6 different types that you get, lactobacillus is in competition with a lot of other ones. So it's not real symbiotic. It's very spore forming and good for preservation. That's why they have a lot of milks and stuff like that. But you'd like to put that with spirulina. Ooh, I reckon, I haven't tried to mix mine. I just use it in certain ways. I don't think it would gel. I reckon if we put both of them in a petri dish, I reckon lactobacillus would win. If I put half on one side, like a little dot on the left and a dot on the right, lactobacillus would spread enough. In the right, you have to put the right medium on that side too, because they're different mediums for both of them. But yeah, it'd be good to hear the results if you give it a go. I bought did you see before the 2 different types, the 3 different types that are very beneficial of the cyanobacteria, and the 2 of them are nitrogen fixing, and the one I got is not. So don't get Arthros spiriflatensis, get the other 2 that were on the, what's the name of them? Fusiformis and Maxima. So Arthrospira fusiformis. Here, I'll just pull up thingo. And this is really good too. They sell this for top dollar at the health food shop. So see up the top here, there's 3 species. This is the one I got, which does not fix nitrogen. It's got a rad amount of chlorophyll. It just looks, and that's what I thought it did. but it doesn't fix nitrogen. But source these two. I paid $50 for my culture. I've had it for a few years now, and it's just still the same culture. It's pretty rad. And yeah, you skim it off the top, and you put it under your microscope and identify it. That's at 400x. But it doesn't, you put it outside and it's an autotroph, so it grabs sunlight and turns it into energy. So, and uses carbon dioxide as well as a electron acceptor, and TEA, electron acceptor, yep. So I don't really do much to it. It looks after itself. So they're saying you should put do all this and that to it. Yeah, okay, if you're going to commercially do it, but if you're just going to do it by home, well, yeah, mine seems all right. Mine doesn't get a virus or it's not sick. It you can see on top of it. I look after it though. I don't mix it with anything, and I don't feed it anything but water. Anyway, enough of that, I think. What does he say? Oh, g'day, g'day. You can grow spirulina in a kiddie pool. Yeah, mate. Yeah. Yep. You sure can. I do it in a 5-gallon pail. It just sits there. It's great. Then I need some. I'll scoop a few out, use it for fertilizer, use it for other things. It's pretty cool. When it gets a biofilm on top, you can dry it out. I got sick of drying it out because when you break it up, you need a large amount to get like a good amount to use it as a supplement, a human supplement. So, PKs. Okay, yep. Raptor, 420 Fred. Hey, Lene, how you going? Nice to see you. G'day. Hello. Raptor says you don't give your spirulina any fertilizers. No, nothing. That's the amazing thing about it. And it's at a good concentration. It's fascinating. Where it says on that page, they got to feed it, and it came with spirulina food. But from my studies, I thought, hang on, why do you need all this stuff? I'm just going to try it without it. And it's how it functions. It's organic, as organic as it gets. Because it's the same with, you see, algae in the ponds. They only use the macro and micronutrients that's in the water, but they still survive even in crap water, in poor conditions, poor nutrient, or it could be saline, or actually that's not a good example because too much salts, they're probably not there. But for me, yes, I don't want to make mine multiply fast. I just use it when I want. It always sits as an extremely dark green water. That's what it looks like. And if I tested it for the ppm, I think it would be, EC would be off the charts. It'd be ppm, it'd be, I don't know, 7,000 or 8,000, I reckon. It'd be extremely high. It's very, very dense. I don't dilute it very much. I just let the rainwater go into it, and that feeds it. And if there's any pollen or anything around, I'll put like a cap or something on top of it. I've got a filter cap that I put on top. So I try to protect it. And yeah, it's a couple of years old. What happened? It might have, no, that was a brown film that put across the top. That was because it was too thick. But yeah, it handles, when it gets cold, it just puts a frost layer on the top of it and it sits below it. So it's pretty cool. Interesting because I've seen other growers use hydroferts on. Holy moly. Oh, mate. You'd need, well, there you go, synthetic and organic. They're into their synthetics. I'm into my organics. Yep, that's no worries. He also recommends the liquid you get off the methane gas composters. Yeah, biomethane is really cool. You can get biomethane collectors and collect them from your compost heaps if you've got enough of it. Or it's more so for your, if you want to do biomethane, you want it's human waste produces more of that. And surprisingly, it's a high amount too. I looked at one because it interests me, because I'm into the off-grid sustainable living type of scenario, and it interests me. And I was surprised to see how much you get from how little amount of input you put into it. But yeah, could you thinking down that line, mate? It fully be sustainable in every aspect. It's really hard. It's pretty labor intensive, but it's so beneficial, so worth it. Like actually, I don't want to start talking about me. We'll stay on topic here. Spirulina seems to be the topic. Free gas and no utilities to deal with. Yeah, kind of. Yeah, well, being off-grid, you still got batteries, and you still got to replace your batteries. So there is a cost on that every couple of years. So if you bring it back, it might be cheaper than what you pay for your electricity, but yeah, there still is a cost. But it's very, very, very convenient if it's set up correctly. Yeah, but spirulina is beneficial. Excuse me. What else can we talk about? Spirulina seems to be on the cards. Should I elaborate around that? Suppose, all right, well, there's not much happening. I'll just show a little bit about spirulina. Unless you please put your questions in or anything like that, so I can get onto another topic or something that you'd like to talk about. It's free floating, yep. Thrives at a pH, yep. Here we go. The autotrophs meaning they make their own food and do not need living organic energy or a carbon source. They get it from the air. So if you want to grow it, though, it's a different thing. See how they want to boost the numbers and really harvest it. So I'm just happy to mine just to plot along. But yeah, if you want to grow it, you can go and do all sorts of stuff. This is what they recommend. I used to grow it when I was getting in. I've done it inside and outside. That's when I used to harvest it. I'd add a little bit every now and then of all the stuff that they gave me. Nutritional. This is advantage for this. Actually, 100 grams of spirulina is a rich source and of essential nutrients for humans, animals, and plants. And it's I'm not going to go into all that stuff. You can see it's an extra good, it's a superfood. That's why it's sold at the health food shops for top dollar. And people have these cyanobacteria farms in aquaculture. 2% spirulina per day. Safely recommended. For your animals, spirulina can be fed up to 10% for poultry. 4%. Oh yeah, because I put a little bit in the pets' cups, and if it doesn't run out in their drinking troughs or whatever, that it'll still multiply inside of there. So when you pour a bit of water back in to top it up, or your catchment tops it back up, your float valve, it'll always be in there forever. So it'll give them a full-on extra boost of micro and macronutrients for your rat animals. And just some safety concerns. 1 or 2 people in the world have had a problem. So, of course, using bacteria or fungi, you've got to take all of these precautions. See if anything else has been said. Have you got a NPK of your spirulina batch? No, but the N would be mineralized and very high because it's very green. So in the chlorophyll pigment has a magnesium, 4 atoms around the outside of a nitrogen atom. 4 magnesium atoms, anions around the outside of a nitrogen cation. So when I'm looking at, that's with the elements that I'd be looking at. But no, I have not got a test done. And the PK, well, the K would be, I'd say, virtually would be extremely low. What's it say on this dietary? Right, I'll see if I can find that out, because that'd be pretty good to know. And I'm sure they got it now. Oh, far out. Oh, look at this answer. What a spin out. They were saying it's a superfood. It's a bit more than a superfood. NPK of spirulina is 10, 26, 26. 10, 26 and 26. Are you blimmin' kidding? Let's see who says that. That's Platensis as well. Dead set. It's a complex fertilizer. Cultivation of Spirulina, that complex fertilizer, simulated by disc Africa. Oh, what? So the highlights: cost-effective cultivation, 1026 fair income. Spirulina conservation is checked that way. Biological conversion of CO2. Okay, very good. Elimination of chlorophyll constants and lipids. Wow. I didn't realise it was so blimmin' high. I'm going to be putting a little bit more effort into it now. Sodium. Anyway, I don't want to read this now. I'm going to read that later. Wow. Now what do you think? Far out. So you're talking for fertilizer before about sunshine, about an amendment in bloom. Well, mate, you're not going to get any higher as for an organic amendment. I'm just going to look at that again, those numbers. 10, 26, 26. 10, 26, and you can grow this yourself. 10, 26, 26. Have you gotten your NPK? The high sodium is the only thing that concerns me. High sodium in it? Oh, I don't know about that. I can look into that. Maybe your Raptor knows a bit more than me about it. Raptor says also that there you go. That's a bloom fertilizer. Exactly. Holy moly. They've got bloom boost fertilizers that you can get synthetically, and those numbers are only a smidgen higher. So I'd suggest look into that, or speak to me later on. Sodium source, you reckon? NPK. All right, let's have a look. I'll go up here for sodium source. I suppose they're saying 46%. Then another one says 75%. Let me share, and I'll go through this with everybody. This is a pretty interesting topic, I think. So up here, Wikipedia, it's probably one of the better ones, 46%. Because most of that's fact-checked. That's why I rely on Wikipedia a lot. Well, 46, so 1 gram per 2 grams. So if you're putting in maybe that's wet. I wonder if it's dried. Let's have a look, see what it says over here. Must have that in here. There it is on the right-hand side. Oh, it's dried as well. So this is out of 100 grams. So sodium, you want to see at least 50 grams. Where's sodium? Come on. Minerals. Holy mackerel, 46%, 45%, 83%. Oh, that's at the daily intake quantity. 1,000 milligrams, that's 1 gram. And that's 1,000, that's 2 grams. Oh, that's out of 100. Oh, hang on. 100 grams. And that's 1 gram. So that's 1% dried. So your daily intake, it's if you take 100 grams of spirulina, which they might recommend, that's going to be 46% of your sodium. That's how that interprets. So, because I was thinking it's not a saline-inducing element, but I love these questions because it's just checks things, gets me to check things too, because who knows? So, if 100 grams of dried spirulina, it's going to have 1 gram of. So, I'd scoop out a cup. I scoop out a cup about 250 grams. So, out of that, if it was dried, actually it's not dried, so there might be a different composition for wet spirulina. Let's have a look at that. Wet spirulina composition. It's got that. So there's only one study. Wet. Oh, it hasn't got the rest of the dry. And it's only a snippet of the article. Environmental applied drying techniques. So it's didn't say wet. So I might have the before and after. That would be good. Materials, chemicals, procedures, characteristics. That was results. Boundary cycle. Here we go. Resources. Okay. No. Results. Moisture, carotenoids, chlorophyll. Righto. They're not really doing the NPK like we wanted. They're just doing that test. Wet biomass. Environmental impact of 1 kilo of spirulina at microbial cultivation levels. Oh, wow. Putrefaction, 10 to the power of 4. No way. Human toxicity, 10 to the power of 0. Oh, because you can't have a kilo. Anyway, let's get back on. Yeah, who'd have a kilo, Jesus. You know, even oxygen kills you. Everything kills you. Too much water. Remember the Boston Marathon doctor? He died in a race from too much water. There's no NPK. Conclusion, oh, they just tested it for that. Oh, well, I tried. That was a good little, see what's been said, actually. Do you think it's like that because they used baking soda when cultivating? Yeah, the different reaction from that. Well, the salts have got to do something. If they're not mineralized, if they're not used as metabolized, they're going to be stored or precipitated or used into transforming into different other things. Depends if they've got an oxygen element attached to them. And if the oxygen goes down in the water, the first thing they'll use is the oxygen attached to the nitrogen, which is still, because the pH they like was the alkaline pH. So that's that still survives with there's nitrates, nitrites even. So if it's I just try and keep a pure culture, so then I don't have any dramas like that. Yeah, it'd be interesting to even test mine, wouldn't it? I didn't end up sourcing those test kits for plant tissue. Ended up getting too high, and the pricing was real odd, and the dealers I was dealing with weren't good. I'm having a lot more luck with the bio-fertilizers. What's Lene say? I take essential ocean marine phytoplankton, and it's extremely salty. The phytoplankton is in sea mineral solution. It's super concentrated. Ah, there you go. So it depends how they culture it. Where people who buy it, they've got to do it on big, massive scales, have the algae farm, no, sorry, the cyanobacteria farms. So that's how they've got to do it, I suppose. And when they dry it, they'll scoop it off the top, and if they don't rinse it enough, it'll still have a heaps of ocean salt in it, which sodium, what's it? I think it's nearly 1% in the ocean, isn't it? It's quite high. And depends where they grow it and their concentration. It might even be higher. Yeah, that's Bleaky. Hello, Bleaky. We're talking about phytoplankton, spirulina today. Essential. It's extremely salty. Yeah, that must be how they culture it then. The stuff that you saw, try getting it from a different source, and they might have a different salt amount on it. But if you want to use that sodium amount in your diet, remember it said before only 1 gram, no, 100 grams of that spirulina has 46% of your salt intakes. So if you were to have 10 grams, that would have 5% of your daily salt intake. 10 grams of spirulina. And I use it as a fertilizer for the plants every now and then. And here's a little tip for you too. If you use a clear pot, you inoculate the inside of it with this, and then it grows on the inside of the pot green. So you'll see the algae growing because there's water and moisture, and then that's going to break down. It's going to mineralize and contain that rad NPK, and then when it breaks down, when the microbes get in contact with it, they will, sorry, it's going to immobilize, because it's those 10, 26, and 26 is immobilized into the cells of the spirulina. And then when the microbes get in, they'll break it down to make it mineralized and available to the plant. So that's a cool tip. I've been doing that for quite some years now. And because I first saw that algae was getting on there anyway, because I've been playing around with clear pots for a while. And I thought, well, why not inoculate it with something beneficial? And then I can grow that on the inside of it. And I've been doing that. Actually, I remember discussing this a while ago, and I showed my experiments with it, with that. Just, it's getting better sources of, well, you've seen the NPK. So if you're into organics, any type of high superfood is hard to get. So, yeah. Yes, I can retitle it something like that, superfood. What else has been said? Hello. Oh, where do you get it? Oh, ocean water is 3.5%. Wow, that's how much sodium's in the ocean water. That's heaps. So that's probably why you're getting a lot in your thingo. When it says perfect to use for lacto fermenting veggies. Oh, okay. Yep. Maybe when it's dried, it might work better, because if it's still active, it might multiply in your culture. But if it's dry, it'll have to meet its favorable conditions before it can regerminate and those spores can multiply. So treating it like a food if it's dried. You got it. Oh, yeah, you're just answering that question. Cheers for that. There's mangrene. Here you go, Main Green. We're just talking about a superfood. You missed out on it. You have to rewatch it. Yeah, it's amazing. Organic NPK is 10-26-26, and you can grow this yourself. He's going, what? Yeah, rewatch it. Lene says, it says chlorophyll wellness elixir under the label. Premium blend of 2 hairline marine plankton strains. Oh, yeah, okay. It says 2. That's pretty good. Oh, excuse me. I wonder if you could reculture yours. So here's something for you to try, Lene. You've got some. So add some to some water and leave it near, and it's pretty warm. So leave it, say, in a cup or something like that, near a windowsill, and see if it starts to grow. And as long as the temperature's all right and the water's not shit, you don't want it chlorinated or something that's going to actually kill it. So get tap water. You got chloramine, which has a lot more. It's got a triple bond instead of chlorine, like we do in Australia. So it's doesn't really. It takes days to break down. So get your water, leave it out for 7 to 10 days, or boil it, or use a little bit of bottled water for that experiment, and tip a little bit of sprinkle half a teaspoon of powder in to half a cupful of water in a clear cup, and see what happens. And I reckon you might be able to culture your own. Okay, you just bought some. But yes, it's fascinating superfood for plants and humans and pets. It's pretty cool. GMF says, well, we lacto ferment at around 2 to 2.5% of the weight of the product to be fermented, i.e. 100 grams of cucumber or cabbage gets 2 to 2.5 grams of salt and enough water to just cover it, ferment anyway. Wow. So you might be able to use ocean water. So that'd be perfect nearly for that culture. Raptor might be onto something. You're saying about his lacto and mixing that and that. It's cool. Awesome. GMF says 2% is also the same amount of salt we add to bread. Wowee. Just a little bit under seawater. That's amazing. And then he says, make sure you dilute it in juice or tea. So salty it makes me gag. Yuck. That sounds full on. Hey, he's a martial artist. There you go, martial artist. Nice to see you, mate. I love it though, thoroughly. Supreme health for you. Yes. Fancy an organic fertilizer, 10-26-26, that you can grow yourself organically as well. Wow, I'm going to get into it more now. That's fascinating. How unbelievably healthy it is. Hear me guessing those low numbers and it comes out and it's blooming high. Geez, good place to check. Read those studies and wiki and stuff like that. That's pretty cool. I love it extremely. I live 30 minutes from the sun and ocean. Wow. Geez, you'll have to go. Oh, you can maybe get the salt water. Cheers. Port Campbell's. Oh, yep. Very nice. You can go and get the kelp maybe and make your own kelp fertilizer. Make it liquid fertilizer. The stuff you get is very diluted. Anyway, stick to spirulina. Seems to be 45 minutes on it, just on spirulina. All right, how about I'll just go and do a few of my little experiments on this. Well, because this is just one topic on it. So I might as well just fully give all the secrets away. Where are we going here? Just find the right section. Plant science, I think. No. Is it that one? I remember because it's under organics or something. That one? can see I haven't done much in it for a while. It's not that one. Oh, fair income. Last folder is going to be, isn't it? No. Fertilizer labs. No. Oh, there it might be. Is that it? No. Still trying to find it. This is not usual. This is unusual for me. I can't find the folder. It's got to be in plant science because it's to do with nuts microbes, actually. I wonder if it's under nitrogen fixing bacteria. I've got a folder there. No, it's not under there. What a shame. There's a few cool experiments that I've done with it. Biopesticides. No, it's not a pesticide. It's not a fungi. Cells. NPK microlection. It was under organics. I remember it. So where's that? Still trying. It's not under that one. Still trying to look for this folder. It's not under the line. Time lapse. Oh, I didn't check study. It's got to be near there. There's heaps of folders. Where's that one? I can't see it either. Oh, there's a thing under it that says My Organics. Okay, I know the name. Look, we can search it. I'll put my little search engine thing out. There's a that's not the right folder. That's on other organic stuff. I mean, I might have to give up here. This is pretty rare. It's the first time. That's bugging me now. It should be microbes. I'll have a bit more look at microbes. There's probably been questions and everything coming through. Sorry about that. Oh wow, put on the chat, Blinky's blooming shared $10. Thank you, Blinky. Wow, that's very nice of you. You have to go and get some kelp from the ocean. Sounds like you're close to the ocean. You can go and get some kelp, and then I can help you make some kelp on a live show. Beneficial never. Just clicking everything now, trying to find this organic fertilizer one. There's pictures of it. that was my fertilizer actually, because I want to show my experiments. There should be a massive folder on it, because I remember I've shown them before here. Because it's been, roots, plant size, that's rhizosphere. Oh yeah, because I've shown them. All right, well this is some of them. I'll go back and see if there's, now that I've found the folder, I can catch up on that. G'day, lucky. It's my Saturday schooling time. Oh yeah, thank you for that. That's very generous of you, Blinky. It's very much appreciated too. Awesome. Sweet. Yep. Yes, thank you. Very much appreciated. So here to do with spirulina, some little experiments that I did. So here's an experiment done of excessive nitrogen on the roots to see what happens. So there's two forms of nitrogen that the plant recognizes, the ammonium and the nitrate. And here's an example of the two. So this cultivar preferred nitrate as for root length and development, but as for the laterals, it preferred ammonium. But here's the concentrations. So in low concentrations, it did this, and medium, it's a lot of laterals, and in very high concentrations, it was probably thinking, yuck, not too good. It was actually sufficient down the bottom here. I should have read the x-axis first, but okay, so they're sufficient. They haven't got extremes. That's a shame. So it only went up to a sufficient. It was foraging because there wasn't much about, and then it went to survival. So it'd be all sorts of abscisic acids. And anyway, so I inoculated some. I can't remember that experiment. This is on because remember I was saying before, on the outside of your containers that hold your medium, if you're using it clear, you can use your spirulina, and when you pour it in a little bit into your fertilizer, into your substrate, it'll pour it around the edges. So it will stick on the edges, and it'll be broken down. See the little green bits there? So it's starting to grow. So if I was to keep this more moist, these little green bits of Platensis, who hold NPK of 10, 26, 26, hold those minerals in there. So they've got them immobilized, so they're not available to the plant. So to have them available, when they die or they're broken down, then it'll be made available on that immobilized element will become mobilized. So it can be taken up by the roots if it's washed into it or, moved and it's got access to it. That's why earthworms and other micro and macro arthropods are handy in the soil because they can move elements and things around. Like earthworms, for instance, too, on the outside of earthworms, it has ammonium. So if it just brushes past the root, we might have put a little bit of ammonium there. But today's spirulina. So it's sitting there, and it's going to dry up and not, so you can see it's not really thriving there. So it needs more moisture, more so, being an autotroph, because it can feed itself from the sunlight. And there's one that had it and one that didn't. See all the lateral roots are a lot more whiter and healthier. Oh, that's right. I remember looking at it now. That was rooted in a medium of spirulina. So all I did was just put a cutting and left it there It looks like it was in bubbling, so that might have been a bit unfair. Actually, no, that wasn't. That was rooted in, and then I put it into the puck, I think, and then it was put in. Rooting for resilience. Yeah, and there's some nice looking roots. See how they're all mainly white? You don't want brown. The brown of this dead bit. You want it to be like that, bleach white color. So because there's a sufficient amount of oxygen, that's why that survived. Oh, mainly, but that's why the roots are healthy. They take up the oxygen. So if it's not enough oxygen, it's when they start to go brown and dry up. I think we need to get that technical about how the root tip cycles nutrients. It goes in through the root, out through the root, exudates, in through the hairs, through fungi. They're also attached and they can transmit it along and through the cells, through the two different ways of apoplast and symplast ways that the nutrients can get in through into the xylem and the phloem. Yeah, look at that one. Not bad, not bad. That's without a flash. So if that was flash, that'd be all bleach white. You see the difference? Good to bad. So if this was probably inoculated with a bit of spirulina on the outside here, down the bottom here, it might still be thriving because there's a good nutrient source for the roots. There's a massive example of what could happen underground from a big massive tree. All branching. It's just fascinating, isn't it? So mainly it sits along the top because see, it's starved for oxygen, but when it's looking for water, it'll go anywhere. So that's probably what these deep ones, they're all looking for the water. And that little tree will get through the seasons. It's got its oxygen source, its water source, nutrients stored in starch it can reuse. Lovely, it's saying. Keep me organic. It's electro. It's a different story. Oh, this must be our roots folder. Okay, I understand now. Yes, that was a pretty good episode talking about how to make and stimulate roots through electricity. And we worked out the different sizes you need to put in. Look at this root mutation. Isn't that awesome? Off the track of it, I was talking about my spirulina experiments that's right. Yes, thank you again for that. It's very kind of you. Thank you. And you, what also throw something, CC's my, oh yeah, thank you. Where you hide the donate link. I don't know, sorry. There's memberships. There's super chat, super thanks. I'm not really sure on that one. Donate. I don't really know, thanks. Yeah, thank you though. GMF says, what AussyCC, so what you think about using fish tank as your, not just your water source, but as your soluble and nitrogen source for your roots. Seems like it could be worth keeping a few fish for nutrients. Yes. Then you'll need to get, a bit about this, about nitrobacter, and you need nitrosomas. So nitrobacter converts the nitrites into nitrates. And actually, no, you don't want nitrosomas. That does something else. That converts the nitrosomas is your ammonium into your nitrite, which your fish pee out, nitrite, NO2 minus. So yeah, to convert that into nitrate, you want nitrobacter. So if you're going to get a fish tank with your fish, get Nitrobacter as well. And that is a rad source of nitrogen, organic nitrogen, as well as your spirulina. So you could probably put in that fish tank a few fish, goldfish or whatever you want, and a bubbler, and inoculate with spirulina. Get one of those 2 of the 3 species that I said before, and it'll fix nitrogen as well. So you'll get a solid 10, 26, 26, and you'll also get your nitrates from your Nitrobacter. So yeah, mate, that's one. That's in growing. It's the next elements outside of the CH and O. From the 17 essential elements that the plant requires is nitrogen, which is 1.5%. So if you can get it somehow ongoing and organically, you've won the battle. So I think that's fantastic. That's a cool little experiment I'd like to hear more about. And I bet you everything's going to be green from here on in. You won't get any yellowing and stuff. I think he says, I'm going to prepare a vape, by the way. Very good vape on. Been contemplating that as well. Have some other species to make the tank stable and self-sustaining. Yes, self-sustaining. That's the way. So I can just keep churning over. So it's something that has to need something to sustaining. Then won't go into it because I want to stay on topic. But what are they called? Little glass shrimp, because they eat the poo. So if you get them in, that'll clean it as well. And then you don't want the outside, so you don't want plecos or anything like that to clean glass, because you want your spirulina to go on the glass. And then once it gets thick enough, you'll scrape at the inside of the glass. It'll all float to the top in these little strands, and then that's when you'll sieve it up in your scoop. And then bang, you've got dry spirulina as well as wet spirulina for your microbes and Nitrobacter to work on. And put a high amount of Nitrobacter in. I'd put in, it depends how much you're going to get, 10 to the power of 7 or 10 to the power of 9 or whatever you're going to get. But if you get 10 to the power of 7, that's 1,000,000. Oh, gosh. Actually, I don't want to work it out now because we'll start on spirulina's topic. But if you do end up doing that, let me know and I'll work out how many grams, because you want it to be a dominant culture. So you want it to really be thriving. But it can easily be that simple. Shipwreck says, I've been wanting to get a fish tank going again. Why not? Everyone else is talking about it. Seems to be the in thing at the moment. Free nitrogen. Plus the phosphorus that comes out of the fish is good too, because of its waste. It's got manure in it. It's poo. Excuse me. And there's also little microbes with the DNA that have been, immobilized, with phosphorus in it too, because I think it's 45%. No, a high amount of phosphorus anyway comes from DNA. That's the backbone of the DNA. So you want to get microbes to mineralize that element that is immobilized in those items, in that organic matter or those fish and things we're talking about. I never understand that. My bad thing. I needed my first vape. First vape, it's blooming lunchtime. Gee whiz. That's good. At least you mustn't be in that much pain. That's a good thing. Shipwreck says, I used to always had one. A fish tank. When I did water changes, I would dump it into the garden and loved it. Yeah, and I had a fish tank also, and I never thought much about the when doing the fish changes every couple of weeks or whatever, with that cool little sucky thing. Oh, Lenane's joining you as well. That's the way. Raptor says, wow, I'm surprised I didn't consider spirulina being grown on the glass of the tank. That's okay. That's lucky why we get into these chats, because I didn't realise that high NPK as well, that people that you made me look up, or the reason why I looked it up was your question, so that's cool. GMF says, AussyCC, so what do you suppose is living in my sprayer? The inside has a green patchy film all over the inside, and I'm assuming that it's beneficial. My water always has a stone running through it with air. That's a cool question, because yeah, I get little biofilms on my sprayers as well, and I think similar. Like on my bubbler where I do my teas for my bio-fertilizers, it's got a film on it, and it's a dominant culture because the biofilm doesn't mix cultures. So that amazes me, and I'd love to do I'd love to plate it, scrape some off, and put it in a Petri under laminar flow hood conditions, so you can just culture up that medium and see what comes out. See what color it grows, because the colors are can tell you the families it is, like Aspergillus is green. And then it's hard to really identify. You can only, like for me, I can morphogenically identify it by under the microscope, so I can see its shape, and then that'll help you fine tune it a bit. But as for knowing exactly what it is, or even from the genre that it is, it's pretty hard for me, because Pseudomonas looks like a tic tac, like Bacillus. So if I was to go and say, oh look, there's some Pseudomonas, it might be Bacillus. So yeah, but I agree with you. It's got to be beneficial, because it's trying to culture under the conditions you've given it. So all the things that you've inoculated or put into that mix, it likes, and it's trying to power on. Go away, everyone else. This is our feed. This is our biofilm that we're going to proliferate in. And the biofilm, they put out heaps of antibiotics as well. It could be, if you use spirulina, it might be spirulina. Sorry about that. It could be lactobacillus if you use any lacto cultures. And I think you get into a few of those, because they are heavy biofilm and in competition with other microbes. Because remember, of the 6 different types, the competitive, symbiotic, co-symbiotic, or mutual, predator, that's 4, predator, and then one's a pathogen, 5. Well, anyway, there's one more. But of all those different types, you want to get the one that's symbiotic, or at least working with your cultures, not in competition, because it might be eating some of your nutrients that you're like NO3, like you want nitrate ones, like cellulase. I don't want to go off about into the microbe stuff. Actually, it was part of the question, though, wasn't it? Yeah, you can get some microbes that break down, are good for compost, and they're the ones with cellulase in their titles. So you'll see them. There's a few different ones. I can't recall them off the top of my head, but those are ones that specifically do that. So they'll break down cellulase. And they might decrease the nutrients that you want to be building. Hope that come out all right. He says, GMF says, like microscopic versions of the movie Warriors. Okay, I haven't seen that movie, but yeah, Shitwreck's laughing. He says, love that movie, and he agrees with you. Yeah, the biofilm. It's interesting getting into the biofilm composition and seeing what they're up to, I suppose. That's a fantastic comment. I only use 12 types of microbes, so I could know my biofilm should be one of those 12 types, because I use them in high amounts, in the billions, with billions. And I've never co-cultured spirulina in with it, because maybe it might be good as a dry amendment to put in there, because the microbes that I got are phosphate solubilizing has those properties in it. They produce phosphatase, the enzyme that breaks down phosphate that's been immobilized into, for instance, dry spirulina that has 26. So to get that out of dry spirulina, you want a microbe that produces phosphatase, or it's a phosp-P solubilizing, to cut it short. And most of mine have properties like that in it. So by me putting the dry stuff in, I wouldn't be... Actually, that's a test. I'm going to do that. I don't have a light in the outside, though. It's dark where it brews up. But I can nearly do a dry test and see if the spores could come alive and compete and put a film on the outside of it and grow on the outside of the container. Geez, I love experiments, because you don't know. It might be more beneficial, and I might get better results. If I did an NPK test of the brew afterwards, it might find it might be a lot higher now, because I've got that culture that's developed on the inside. The biofilm's being created there. Fantastic. Yeah, I appreciate your comments in chat, dead set, because it gets my brain going for different experiments. It reintegrates my education, and it's just blooming. I really like it. I get a kick out of that, and I appreciate it. So I just want to show that appreciation to everyone in the comment. All these comments that everyone's saying, that just different things about different situations gets my brain thinking. So it's cool. Even the comments that you might think, oh wow, they're a bit whack. They're not whack to me. It just gets my brain thinking again. Oh, that's what their thoughts patterns are. Oh, okay. How can I elaborate on those, adding that into it? Like microdosing with a Dino Vape 7XL. Sounds like it's pretty big. 7XL. There'd be no biofilms in that. And that's one interesting thing about the biofilm, where they want to proliferate in. It won't be low pH, not acidophiles that we're into. Oh, actually, I better not start going on down that path. You know me and microbes, I can easily take up another hour. So write in your comments, is there anything else that we can talk about today? Because it's about a 30-second delay. Because otherwise, yeah, I don't want to talk too much about microbes. I'll try and some of these cultivation of spirulina, the NPK complex fertilizer. I'll put that study into today's show. So if you want to go back and have a look at it, that's where you can get that one from. I'll see if, there was one other study done. Yeah, to get that 10-26-26 complex fertilizer, NPK, and you can grow it yourself. Wow. For all those people in weed nerds, yeah, you might be lucky. Because, all you got to do is keep it wet. Well, I don't know about dry inoculation. That's what Lene's going to do, that test with her powder. And I can do the test, I suppose, on the inside of mine, because I got some odd dried mine in the past. But yeah, to keep it wet, to inoculate someone else, all you got to do is just give them a little bit of the batch and grow that. So it's even just a shot glass full, half the size of that is enough to put into an ice cream bucket with some fresh water and under the sunlight, and you watch within a couple of weeks, it'll be going green. Actually, probably within 7 days, if it's in 7 days of sun, it'll be all pretty much a light shade of green. And if you put that up to the hue index of nitrogen, you'll see that, yeah, there's some nitrogen being assimilated in there, and it's been stored and immobilized in the cells that you've created. Pretty cool stuff. And it's all for free. This is all for free. Just by doing the old organic thing, you just got to put labor in. And there's not much with spirulina. Well, my culture's been fine for a few years now. Well, it probably hasn't been fed for at least 12 to 18 months. I haven't put anything else in there besides water. There's elements in the water, of course. There's NPK and macros and micros in water, even in rainwater. So it still sits in there. It looks very healthy. labour and start with a pure culture. Yes, well, that's what I did. I got well, with this one, it must be it must be biofilm producing, because other microbes in the air, there's heaps of fungi and bacteria cells in the air that just travel straight into it. It gets blown on the top and they don't proliferate, they don't grow or nothing. And I've done this in various locations too, with different pollen and different air conditions. So I think it is a heavy spore related, and it's not real symbiotic in that sense, just from thinking about that. Because otherwise, why haven't other microbes got in there and done their thing? I think it did get a bit sick a while ago. I can't confirm that, but I think my bacteria got a virus, because I saw some other things going on. I can't really go into it, but yeah, because there was a brownish biofilm, and it's usually a greenish biofilm. I thought, oh, okay, but yeah, it got over it and it's healthy, and I didn't have to. I can't remember babying it back, if I fed it or not, to bring it back in. Gave it a bit of a boost of something. But in general, it's pretty much looked after itself. But yeah, starting with a pure culture, you can't go wrong, because you don't want to be culturing the wrong thing. Who knows if they've got actual algae? You can get some, the spores of algae are carcinogenic. The blue-green algae, some of them, there's bad ones as well. And there was a lake in the middle of Australia. I remember seeing the story that the residents around the outside, they didn't know how they got sick, cancer. And it turns out it was from the spores of the lake. It would go green from the eutrophication from the runoff of fertilizers, would run into this lake, and that would exponentially feed the algae, so it would get it to grow too fast. And when the spores would come up, they would grow and they'd come in our bodies, and it wasn't healthy, that particular species that it was. So by getting a pure culture, you can't go wrong. You know you're getting what you're getting. And I got a pure culture when I started, but I've handed it out to people, so at least they know what they're getting, I suppose, too. But they'd have to trust me. So yeah, it's hard to trust people with what's going on these days. But you can't. That's a good point. Start with a pure culture with anything. Look up the Dynavate, no worries. I had 3, 4 of her. Yes, it is a shame. New laws have changed in Australia now, where you can't get vapes anymore. Believe it or not, you just can't get them. They made it illegal. So I don't know if you can still source. They say, the Stalks and Brickle said to me that you can still get the medical, the Mighty Plus and the Volcano Medic, but everything else is illegal. And when it was asked in Parliament too, the answer was, what did they say? It was so pathetic. Oh, that's right. Health doesn't mix with border protection. Thank you. Or sorry, the border protection doesn't mix. The rules don't apply to health, or something along those lines. Or it was a 5-worded sentence that was just, that border protection's like the border, the security stuff. They don't talk about it in the news, because you just don't. That's what they reckon. So it's kind of like a wash-off like that. It was good luck, Australia, again. Not enough money's been made, so I'm not going to get into politics. That's just what they said. You don't know what I want to think about it. I don't swear on the show. What are you saying about that? Oh, yep. They can get... Yep, that's right. I reckon too. That really... Yes, it is a shame. Well, they're not putting health first, so it's they didn't have to bring dry vapes into it, like wet vapes, where the kids are getting into. Fair enough. The kids aren't getting into dry vapes. It was just another excuse. They just throw it into the list. So yeah, Nothing much can be done, eh? Unless you're a politician. I'm far from it. I'm not working with... Anyway, so spirulina. There you go. Whack your comments in now, because it's been going for an hour 20. Raptor says, yes, you're using a dry herb right now. That's right. They are medicinally needed for medical patients. Otherwise, they're going to get people going back to the bong and having water on the lungs, which I can... Oh, yuck. That's why I turned to a vape, to get rid of the water on my lungs. So that's all Australia's doing. They're just going to get you back to the bong again. So people's hoses will be getting cut again and all that sort of thing. That's all I'll see. There's no whinge about that and blah, freaking blah, politics. Dead Ned. Okay. You can make... Yes. Yes. It's going into politics stuff, and yeah, it's always a negative thing. It doesn't really win, does it? Especially in Australia. It's all right in Trump land. That's what it'll be soon. But all right, spirulina. So put in your stuff about anything to do with that again, and maybe what else we were talking about. That's really all today is. I'm just going to reword it about spirulina, how its benefits for humans and for plants and animals, and what it does. And it's, oh yeah, and then I'll put in too that NPK 1020. I reckon I'll remember that for the rest of my life now, just because it got me, because I'm into looking for organic sources that have immobilized elements inside of them, so my microbes can break them down. Like goat manure has 1.3 is to 1 is to 3. Chicken manure has 5 is to 2 is to 1. Alfalfa meal has 2.5 is to 1 is to 2.5. Cattle dung, they reckon, 0.35, 0.12, 0.17. That's what that study reckons. And, okay. So yeah, I've been just doing a little bit of NPK organic input stuff myself. So to see the 10-26-26, I'm going to probably halve my batch and try and multiply it and put it in my feed program a lot more often. Instead of just giving it a once a month boost, I'll try and maybe put it into a weekly thing. Just tip a 1 cup into a mix of a bit, and yeah, that's fascinating because I only use 3 types of nitrogen fixing bacteria at the moment, but I'll be having 5 when things are good. So at the moment, I've got to look for nitrogen sources instead of getting it out of the environment from my microbes. What's this? I have a Pax Plus, not loving it. Good looking others. I wish I could afford the 90 Plus. Yeah, they're not cheap, eh? $500. It's just crazy. But if mine went, I'd probably have to pay it again because I got a Pax, the first one, and I've used some other ones, and they're good, but they just don't quite get me to where I want. Raptor says, how long does it take you to get to harvest your spirulina? Under favourable conditions, if there was, I think every 10 days to 2 weeks, I was doing, I can't remember the numbers, but I was scooping off the top of my pail. That was when I was giving it boosts, when I was in grow mode, not in like normal mode like now, where it's just auto troughing out of the sun. So I might start and feed it. Actually, no, then what would I feed it? Because I want to keep things organic. Anyway, the question was, how long does it take to get a harvest? I used to harvest more every 10 days to 2 weeks. So I used to pull off the top. I'd have a little container where I'd sit it in. So I'd put a scoop under the water and scoop it off and then tip that into a container. And the scoop would be, it wasn't a fish scoop because it would used to clog it up. It was, what was it? Not a piece of, was some sort of plasticky thing with only a few holes in it, and you collected it up, sort of forked it to the corner. It was a fork. It was a fork. So you fork it all to one corner and then pick it up with a fork and then put that into the bowl and then dry it out. It takes a few days to dry, and then it dries extremely dark purple, and then I just put that into my bags and collect it. That was the harvest process. Yeah. And now I'll be putting in, if I can get some more containers. Yes, that's probably the problem, is the containers. But what I plan to eventually, if you've got any white containers outside or white see-through plastic drums, you know, agricultural molasses comes into it, and 1,000-litre drums, for the 250-gallon ones, they're square. That's an idea to do it in. So you can put a hose at the bottom, connect up your irrigation, and yeah, when you have feed it water, you can feed it some of that. Keeping in mind, though, that it might get too thick and might clog up your emitters. But I shouldn't be telling you what my thoughts are anyway. How long does it take for you to get a harvest? About that. And people that have done it over big, large scale, I think they'd have a feed program of every couple of weeks too. In winter, it wouldn't be as much as in summer because the temperature is into its favourable conditions. It likes around about 30 Celsius or like a bit over 80 and 80F, and then it grows well, and then it multiplies, and it's that filamentous bacteria cells, so they just keep joining onto each other. So to get them to keep joining, the temperature is a humongous thing of it. So you can go and feed it whatever you want, but if you haven't got the right temperature, good luck. So that will do about that. I just remember a jar of green powder. I have it includes spirulina. Yes, yeah. Don't get rid of it. I could mix that in water and use that too, couldn't I? Read on the side of it to see what else is into it, because it might just not have that one culture. It might have a few other species of other ones that you don't want. Like Raptor said, you want a pure culture if you can, but if you're going to try and wing it, make sure you see what else is in there, because if you're going to get something else that's going to go a green sort of biofilm on the outside, you want to make sure it's the one that you want, if that makes sense. But yeah, there's nothing wrong if you could try it, like that Linnaeus experiment that I mentioned earlier, for sure. I think it says, I bought a lot of SS thermosis from LD. Okay, that's the good. Or Barry Humphreys, Standy Sown. Okay, sweet. What can we feed the spirulina culture? Let me enter that with a slide. You'd want baking soda, potassium nitrate, or sea salt. There you go, your sea salt again. Potassium phosphate, and then iron sulfate. So that's to get its macro and micronutrients for growth. But if you just want to, so in winter time, it might be too cold. See that's the temperature you want here? Oh, mid-80s. Okay. So if you're going to go and feed this in winter, it might get to, you might make your culture sick because overpowering it, and it's too much nutrients in the substrate, and it can't use its metabolite. It can't metabolize them because its temperature is too low. Because the chemical reactions and enzymatic reactions are, it needs temperature to speed them up and to get them functioning. So yeah, that would be when you're around these favorable temperatures, you could go and add this to it as an example. It's not real organic, but it's sort of I think it'd be hard to get an organic feed for them. That's what I was thinking before, without actually adding elements. It's kind of synthetically, because these are synthetically produced, so you can say they're synthetics. Sea salt could even be synthetics these days, with some of the way that they produce it with chemicals and stuff like that. Or if they're going to house it like you see in the picture here, there's probably could be organic because there's no real additives to it. It's just an element. That's my understanding of the organic and synthetics. So yes, there's one recipe anyway. I'd be, if I was going to get into it, I'd just put in the search engine spirulina growth recipes and read a few of these studies, because these are the ones where they've done under lab conditions with exactly the things that they say to get the results. Where if I was doing it at home, my water mightn't be 0 ppm, it might be 15 ppm. So those 15 elements could be something that's going to sway the experiment. So by me reading this lab studies that's being conducted, it gives me a good understanding of what's going on. Yeah, see, there's heaps of other microalgae. So you might find that in your mixed cultures that you buy in powdered form, you might be culturing up some of these examples below. There's a lot of them where you only want the spirulina. SPP means species. Chlorococcerum. Just reading some of them. All right, that'll be good. Stop sharing. Did that help with the recipe? Was that enough recipe information? Can you let me know, please? It was a good start. Did I answer your question, mate? Blinky, it's got kale, wheatgrass, barleygrass, and monk fruit, as well as spirulina wheatgrass. Oh yeah, sweet. Well, if that's pretty much all it's got then. But see, on the outside of those grasses, you don't know what's on those. They're not likely to be grown with spirulina though, but they could have another type of microalgae if they're grown under wet conditions. They might have, if there's micro grasses or hydroponically grown, they could have little bits of unwanted algae, blue-green algae, not the bacteria that you want. You could tell. You could look under the microscope and see the cell types. So you'd have to see if one's prokaryotic versus, I'll put prokaryotic versus eukaryotic, and then I'll say microscope, and then there should be some pretty rad images here. Oh yeah, I'll just go back and share the screen. So how to identify it under a microscope? If you've got a microscope that goes down to 1,000, you'd probably only need, even 500 might get it. Well, here's kind of one. I was hoping on the live one. There is it there, but the organelles inside of it you won't see. See in here, you'll see cells, like up here. That's probably a bad example too. Because this one, see, it looks like a Tic Tac. That might be Bacillus or Pseudomonas. They're the same, like that Tic Tac shape with its flagella at the edge of it. And that's the prokaryotic bacteria, or spirulina, where the algae is over here, and that's got all its organelles inside of it, and you can see it's all its different action, and they're totally separate. So this up here is eukaryotic because all the cells are joined, and the prokaryotes are single-celled bacteria, like on the left here. Oh yeah, left over here to right. See how they're all single-celled? So that's some coccus cells. All right, that's That's how to look under the microscope and have a look. Where I'm up to. Oh, yeah, I need to find the organic sources. You're similar to me because I'll be coming up with that too. So what you'll need then is nitrogen-fixing bacteria, for instance, to give it its, well, it gets nitrogen from the sun, but it's other elements. So you want to somehow produce phosphorus without being synthetically done. So you'd have to have an element with phosphorus in it and then have a microbe break that down, making that element available, and that's how it's done. So how would you'd have to pasteurize. If I was going to do it, I'd pasteurize something with phosphorus in it. It's high, langbonite, sterilize that, pasteurize it, clean it, get rid of the microbes, and then I could put it in there, and then that would have the elements inside of it that's organic. And then I'd have to put microbes in there that could break it down if it didn't get broken down itself. That's nearly a topic in itself, Raptor. Organic sources for spirulina. That's just about a topic in itself, because I'd like to really look into that. Well, that's probably why I'm stuck with feeding them the air, because I did it a while ago and reached a wall. That's a great question. I love these questions. So it's 100% organic. Oh yeah, sweet. That's pretty good then. That's a good start. At least it's organic, but you don't know if some of those have been grown, microalgae have been hydroponically in the mixture, if you know what I mean. So you'd have to somehow isolate that culture. It's worth a try. Who knows what might happen? And then you might be able to identify it from morphogenic expression. Just by looking at it, you can see this is going to happen, or this, or if I add this to it, it shouldn't be right. Or if you pH it, the pH of spirulina is 8.5. So when you test it, it might be 6.5. So you know that your culture is not the spirulina culture that you've got, as an example. So these experiments, they all prove something. Organic or low sodium. Well, what about just chlorophyll then? But do you want to NPK it, or do you just want a nitrogen source? Blinky says, my niece and nephew are hopefully on their way back to Australia. If the IT failure, yeah. Oh, and they got a microscope. Very good. Might be able to get fully scienced up. My childhood, I buy cottage seed and eat the produce. Okay. Very good. It's an hour 38. Fair income. It's going to we're going to have to, I know you want to go and do something too, so I'm not going to hold you up. If anybody can remember that question that Raptor, organic source for spirulina. I love that question. For me you could see me go through live all different papers and different stuff like that, and how I would try and get an answer. I reckon that's rad. So I try and remember that question when I'm stuck in a few weeks' time, or probably next week. Yeah. And thanks again, Blinky, for that $10 support. It helps. And it's very, very nice thought. Thank you. Because I try, like I said at the start of this too, I try. YouTube's got a different approach to their monetization, where if you have a EPSA approval, which is education, something, and scientific approval, you can talk about bad topics, and because you're trying to promote and tell people the bad things about them as well as the good things, and that's what I've been trying to get for forever. So yeah, I'm trying to reword things and not swear and all this types of stuff, just to try and satisfy their needs. So one day the show might be monetized and I can get a result out of it. I do enjoy it though, because there's really good banter, and I enjoy the good topics that's been talked about. It's fun. Where I was up to. Have a good week. Thank you for that. Thank you for your support. Blinky. Supreme Great. Hello, mate. You're just right at the end. Thanks for the info. Oh, you might have been listening. No probs. Raptor says, well, makes you think, how is the organic spirulina being sold for health is being made? Oh my, that's interesting. What a fascinating question. How's the organic spirulina being made? What I reckon is, what I'd do, if I was approached to that while we're still on the spirulina, oh look, Blinky is a new member. Thanks, Blinky, for coming in, member. There's some, if you look under the members' rights, there's a few members things you get for different levels, and you can see there's a few videos that haven't been released to everybody. And there might be some playlists too that you get bonus with for being a member. Thank you for that. If someone approached me and says, I want to organically feed that, my spirulina, I'd have to produce the elements required, and then say in a fish tank, and then that would be the feed for them. So I might have quite a few fish tanks producing different elements in it, and it might be, one might be high in nitrogen, one might be high in potassium, phosphorus, all that sort of stuff, and then I could drip feed it out the bottom. That's how I would achieve that. So I'd take each tank separately. So, all right, my nitrogen tank, how do I do that? How can I get immobilized high nitrogen sources turned into mobilized nitrogen elements that the plant requires, or the source or the microbe what I'm feeding requires? Because that microbe might be able to assimilate nitrate. For instance, spirulina, it might be able to use nitrate as a nitrogen source where plants can't. So I'd be taking each element on its course, on its due course, and looking at that. That's to summarize that question. Summarize that humongous question, which is a rad question. Yes. Thank you, Fred. Yes. Blinky become a member. Very good. Thank you for that member stuff and the support. I thank everybody for the support. Even just being in chat with these rad comments is support enough, let alone the financial benefits. So good on you. Because everybody in the chat's just terrific. Look at nearly 2 hours again, and this has only been spirulina, fed into only spirulina. And that's not even a topic that I really get into. It's just a topic that I suppose of it's a study area of mine. So actually, I suppose I could have got into it then. But it's not my key focus points. Yeah. But it is a form of fertilizer, bio-fertilizer. 10, 26, 26, that's going on the big headlines. So I'm not going to go on anymore. Save your rad questions for next week. Thanks, Sunshine. Again, save all your questions for next week, and we can have some cool chat and banter again. I appreciate new members and the support that they give me, and everybody for just turning up and just saying g'day in chat. And even those people that don't say g'day, I know they're sitting there listening because they just want to learn too. So good on you. There's nothing wrong with that. Good stuff, man. Thanks, Shipwreck. Stay warm. Raptor says, cheers, everyone. Good chat. Yes. Thanks, everyone. Have a fantastic week. We can do this again next week. See you again in 166 hours, odd, because this has nearly been a 2-hour show. Yeah, so it's so rad. So that's right. I'll say goodbye and then sit there for 30 seconds afterwards because cool comments come through. So thanks again. Happy breeding, happy growing, and good health to you all. I'll see you next week. Yeah. Thank you. Thank you. Thank you. Thanks, everybody. See ya. See ya. See ya.